inviting me to give this talk on different designs in eye tracking studies. I hope that we can a little bit interact with each other, not only in chat, which I cannot see right now, but I guess that the organizers gave all us the possibility to unmute ourselves. So during my talk, I'll ask simple questions, easy to answer. So please do not hesitate and help me answering my questions. And also, if you have any questions that are like, immediate questions, do not hesitate to ask, because the lecture is for you. And I know that from Krzesi's presentation, the opening presentation, that we are a diverse community, interdisciplinary. And for some of our participants, probably what I'm going to say will be ground truth, but for the others, there will be some novelty in it, especially probably the methodological part of the talk. So I must admit that I do not have an easy task, but with your help, I guess we will build some knowledge in eye tracking. And during my talk, I would like you also to think about your potential eye tracking projects, because the aim of the talk is to help you to design your eye tracking little projects during hands-on session. This is for those of you who will participate in the afternoon sessions. And so let me begin, and let me begin with stating that, with agreeing with the final remark made by Pivy during her excellent keynote, that and also question about individual differences, and that eyes can be like a fingerprint. And it's true. We might not even think about it, how much information can be provided by just looking into someone's eyes. And this is my first question to you. Do you recognize the owners of these eyes? The AI, what do you think? It belongs to what kind of animal? Any ideas? This one.
Fish.
Thank you, Tomasz. The points goes to you. Good answer, fish. The second one, the B one. This is easy one.
Chameleon or something?
Exactly. C.
Eagle, hawk.
Eagle, hawk. And the last one?
Octopus?
Fantastic. It might. I don't
How did you know that?
But octopus, yeah, Jaroslav?
Yeah, I think now it's an octopus when he said it, but to me it was a frog, eye of a frog.
Okay, so the second thought was good one, is an octopus eye. When we look at human eye, we can easily decipher gender. So I guess this does not have to be even a question, that this the top eyes belong to young women, whereas the bottom ones, probably girl or a boy. Probably boy, younger than the lady above him. And so by looking into eyes, we can tell many things about people. And what can we tell when we look at eye movements? So by analyzing movements, we can try to decipher what a person is doing. And we also would like to know something or tell more about someone's mental states. And can you tell me what the person is doing?
It looks that the person is reading.
Exactly. Thank you, Lena. The person, and another question, is it a real person or is it avatar?
It's an avatar.
Thank you, Lena. Lena, this is true. This is avatar's eye. And if you would like to know more about how to make avatar's eyes looking more like human eyes, you can refer to the work by Andrew Dochowski, who is going to give his talk, I don't know, in tomorrow or in 2 days. Great work on trying to modeling eye movements by adding a little bit of jitter and pink noise to avatar's eyes. And what eye tracking is doing, what we're doing when we tracking someone's eye movements, we look at the sparkle in their eyes and make it relative to the center of pupil. But I will tell a bit more about it later on, just to make sure that we all start from the same page in terms of eye movements in visual information processing. So imagine that you are asked to look at this painting and you freely explore it. So from eye movement perspective, you make a sequence of fixations and saccades. And fixations are relative stops of eye movement. Why am I saying relative? It's because we know that even during fixations, there are small fixational eye movements, and there is quite a bit of research right now on, for example, microsaccades and cognitive load. Fixations allow to stabilize image on retina, and we measure the number of fixations, durations, and in scan paths, and this is an exemplary scan path over here, fixations are visualized as or depicted as circles, and the relative size of a circle represents duration of a fixation. So fixations are intermixed with saccades, and saccades are really rapid eye movements, and they help us to move our focus to a new location. They are characterized by relatively high speed and also amplitude of the saccades differ depending on what we are doing. For example, if we just freely explore something, we can observe dynamic between the size of fixations and size of saccades. At the very beginning, we just briefly move our eyes around the picture. So small fixations will be followed by longer saccades. Whereas when we find something interesting to explore, we change the way we move our eyes and we make more stops. So longer fixations are followed by shorter saccades. There is quite extensive study or research when we analyze the dynamic and how we change the dynamic from more ambient focusing toward more focal one. Coming back to the main topic of my talk, how we design eye tracking study. Before designing eye tracking or any study, we need to make some crucial decision. First, what we would like to study, and then who will be our participants, so our sample, and how we actually going to study. In the first example I'm going to describe here, I thought about studying perception of emotions and compare it between how deaf and hearing individuals perceive different emotions, whether there are any individual differences. I'm referring to a question asked to Heidi during her keynote. And how am I going to test potential differences between deaf and hearing individuals in perception of emotions? And I'm going to do it by representing different faces expressing different emotions. Do you think there should be any differences in perception of emotions between deaf and hearing participants?
Yes, they will be.
Any idea behind it? Why do you expect those differences?
It's the way they perceive information, and the way the deaf people understand emotions is different than the way hearing people understand emotions.
Yeah, there is also this theory of compensation that because hearing impaired participants do not have this audio channel available, so maybe their visual attention is strengthened or enhanced. So before I show you results from this study, we need to hold our horses and learn a little bit of methodology. So I concentrate on the how part of our making decisions. So different research designs. Basically, there are 3 types of them. The first one is a correlational design. And if you have any 2 variables measured, you can correlate them. And this would be correlational design. You just look for relationship between at least 2 variables. This could be, for example, cognitive load and pupil dilation. And you check whether, depending on the level of cognitive load, there are differences in pupil dilation. So the higher the cognitive load, the more our pupil dilates, for example. Quite extensively used another research design is a natural groups design, and the example between deaf and hearing participants would be a nice exemplification for this type of quasi-experimental design. So you can, in the literature, you can see 2 different names, either natural group design or quasi-experimental design. Why quasi-experimental? Because it resembles a true experiment, but in fact, it is not an experiment because we are not manipulating our independent variable. So if you would like to check differences in visual attention between groups that are naturally occurring, like for me, from my perspective as a psychologist, this would be some kind of a clinical group referring to a sort of control group, for example, depressed versus non-depressed. Why? Depression, presence of depression or no depression would be my independent variable, but I do not manipulate. I do not tell who's going to be depressed for the sake of the experiment. I'm just testing the differences. The same for testing people with hearing impairments versus people without hearing impairments. So this is the main difference between quasi-experimental and experimental design, which means that in experimental design, we have independent variable that we manipulate. And I'll show you different examples of eye tracking studies when we manipulate either the task that is given to our participants or time of measurement. With experimental design, so this is the key feature that distinguishes natural groups design and experimental design is the fact that we what psychologists are believed to like, we can manipulate with something. We can change conditions in which our participants have to solve a task, for example. And it would be good if we can control for any confounding variables. Just to provide you something that might be handy, this is a decision tree that helps you find out what kind of a design you would like to have for your study. So the basic decision you have to make is what is a type of your independent variable. independent variable can be treated as a predictor if it's quantitative. So by knowing something about cognitive load, I would like to check how pupil is behaving. And this would be correlational design. Whenever you have qualitative independent variable, qualitative means categorical or dichotomous, like in the example with deaf versus hearing participants, or depressed versus non-depressed, or depressed versus socially anxious, for example, then we need to make a decision. So if we have natural groups, then it's a quasi-experimental design. But if we, for example, like to manipulate with cognitive load, and in one group will have no manipulation, and the other one would be asked, for example, to count down by starting from the number 300 and count down by 17. This would be kind of a demanding condition, and we introduced this as a manipulation. Something that I missed to say before is that with experiment, you need to make sure that you randomly assign your your participants to different groups, to different conditions. For example, solving different tasks, easy, medium difficulty, and difficult task. With experimental design, there is another decision to be made, how your independent variable is introduced to the study, whether it is a between subject, between groups, independent variable, so each participant will have only one level of independent variable influencing him or her, or whether we have within subject, within group manipulation, that, for example, people will be solving different tasks, tasks of different difficulty, all of them, not just one. And most often, quite easy to find, an experimental design would be a mixed one. Mixed experimental design have at least one between group independent variable and one within independent variable within group independent variable. And I'll tell you in a second why it's good to have a mixed design. And mixed design is a complex design. We tend to say complex design when we have at least two independent variables. And having two independent variables at the same time allows us to test main effects of each of the independent variable, and also interaction between them. And I'll show you examples of interactions. So coming back to the first example of natural group design, the one in which I was interested, or we were interested to test differences between deaf participants and hearing participants, how they perceived different emotions. So answering the question, who am I going to study? We decided to compare, to check differences between deaf participants and hearing participants in the way they perceived emotions. And over here, you've got an example of scan paths from a deaf participant, and a hearing participant. What we observed is that deaf participants have less fixations and longer fixations. They're like less exploring the face comparing to hearing participants. So my first independent variable in this study would be group, and it has two levels here, because I'm comparing hearing group and a deaf group. and this is not manipulated variable, and it's also introduced between subjects because one can be only either hearing or deaf participant and not both at the same time. And so then how am I going to study perception of emotions among deaf and hearing participants? We did it by presenting faces. For simplicity now, let's say happy versus angry faces, but we can have other types of emotions like sadness or the one that it's depicted over here. I guess you see that the face is now slightly smiling or neutral, but the task for our participant was to recognize emotion, which is dynamically changing. So I don't know if you be able to unmute yourself that fast to tell me what is the emotion as fast as possible, what type of emotion the person is expressing.
Surprise.
Okay, so this was the task for our participants. As quick as possible, try to decipher, decode emotion that is presented, and the emotions were made by morphing neutral expression with emotional one. So we were measuring recognition time, and also, of course, we were tracing or monitoring eye movements during recognition of emotions. So the type of emotion is my second independent, and this time manipulated variable, because I'm choosing which types of emotions will be presented. But to save or to save my effort in finding participants, we decided to introduce this variable within subjects, which means that both deaf and hearing participants were presented with morphing faces of expressing happiness or expressing anger. So so far, this experiment has two independent variables. First one is not manipulated, the group, hearing and deaf, and the second one is the type of a face, happy versus angry, or as you nicely decipher, surprise face. But although the study was not tested during the pandemic, I found it quite up to date, given our current situation, and that in most places we need to cover our bottom part of face. So we introduced a third independent variable, again also manipulated. The first level of this independent variable you saw before the full image of face was presented, and we also decided to present half of the face, and also trying to figure it out who will be better in recognizing emotions or faster, deaf participants or hearing participants. So try your chances right now and tell me what type of emotion is expressed here.
Sad.
Yeah, exactly the person. This person was expressing, wait, I'll try to expressing sadness. But as you saw, it's not that easy and quick to recognize an emotion that is half visible. So to summarize, so far, our study has 3 independent variables. Each of the variable has 2 levels. This is a inner nature variable that in order to name some factor, a variable, it has to have at least two levels. And our design is complex because we have more than one independent variable. And the first one was, as I mentioned before, introduced between subjects. So we had a group of deaf participants and hearing participants. The second one and the third one was introduced within subject, meaning that all of our participants were presented with different emotions expressed by different individuals, of course, because we have couple repetitions of the same emotions. Visible, the full face was visible, or half face was presented. This type of a design, as I told you before, is called mixed design because we have one independent variable that is between subjects and two, in our situation two, but it's enough to have one within subject independent variable. The design of study can be complicated in two ways. You can either add more variables, or you can add more levels to your independent variables. And I'll present you results of our final study design. In our study, we decided to add another group that we thought is potentially interesting to contrast with deaf and control participants, namely socially anxious participants. And if you look at the literature on social anxiety, you may find that when, of course, assessing attentional biases, you may find results stating that anxious participants are faster in orienting their attention to negative threatening stimuli. So they produce some kind effect of early vigilance to threat. But also once they spot something threatening, in many cases, they have difficulty in disengaging from negative information. So we thought that it would be interesting to see whether they will show which of the effect they will show rather in our study, whether they will be more vigilant, so they will be quick at deciding what type of emotion is presented, or whether they present rather the difficulty in disengaging from emotional information. And so the final study design was 3x3x2 mixed design. The first three referring to our first independent variable, meaning the group, anxious, control, and deaf participants. The second independent variable was the type of emotion that was presented, and we decided to add sadness. So we compared happy faces, sad, and angry faces, how emotions were decoded. And the last independent variable in this example was the face visibility, whether it was full or half visible to our participants. And what can we see from the graph? And I hope it's in line with your expectations. One of you mentioned that deaf participants should be better in recognizing emotions, and by analyzing recognition time, this is the tendency that we can observe, that both for full or half faces, when we compare the three bars, we can see that for all of them, deaf participants tend to be quicker in recognizing emotions, whereas anxious participants show significant differences between between deaf and anxious participants. We could see that anxious participant presented rather difficulty in disengaging or recognizing facial emotions. When designing eye tracking study, sometimes or most of the time, you present different stimulus at the same time, or you would like to, even with face, you would like to check how much information, how much attention is devoted to different part of the face, whether we recognize, and this can be this can be seen when we look at the recognition of happiness as emotion. When the face was fully visible, the recognition time for all three groups was significantly shorter than when mouth was covered. Then the recognition time was prolonged significantly. So when designing studies, sometimes we also, most of the times, we are interested in differences between different areas of interest, and we can treat areas of interest as another within subject independent variable. Just to give you an example, a face expressing happiness can be analyzed as a whole. So how much time was devoted to the whole image of face, but we can also analyze part of the face by drawing different areas of interest for eyes, nose, and mouth. And we can then calculate eye tracking indices, and I'll tell you in a second what these can be, and compare how much attention when recognizing, for example, happiness or recognizing sadness is devoted to eyes versus mouth. When we recognize happiness, it's mainly due to our smile. So probably the proportion of time for happiness would be higher when we look at the mouth, comparing eye indices. You can also, instead of presenting one stimuli and divide it into different areas of interest, you can present different emotions at the same time and test for attentional biases toward some of the emotions. Over here, you've got a typical set of stimuli for an eye tracking study in which you would be interested in testing attentional biases. And in our study, we were testing depressed participants and healthy participants or without any dysphoria would tend to look more to happy faces. Whereas when you test depressed participants, for example, they do not show this typical attentional bias toward positivity. So they would spend equal amount of time on looking at each of the faces, or they would show a mood congruent bias and look more to sadness over here. In literature on attentional biases, we tend to calculate proportion of time spent looking at each of the stimulus. So for example, when we analyze first fixation duration, we can calculate a relative score comparing, for example, the time of spent looking on positive face, on happy face, in relation to all other faces. And then if you have 4 stimulus, then around a quarter 25% of time would be equal distribution of your attention toward all four of them. But if you have a biased attention toward, for example, dysphoric or happy face, then the number would be higher. And by drawing area of interest, you can then calculate all sorts of eye tracking indices for each of the area of interest. And for example, we can just to compare how our 50 participants were visiting or looking at angry versus happy face, we can calculate the total viewing time. This would be dwell time. So out of the whole presentation time, and the presentation time over here was 10 seconds. So how much time participants spend dwelling on angry face, comparing on dwelling on happy face. 24% of the time they were looking on happy face, comparing to 20% looking on angry face. We can calculate number of revisitors, how many people from our sample actually came back and look again at the angry or happy face. What was the first fixation and average fixation duration? How many fixations they made when they were looking at each of the face? Also entry time. This is really good indicator of initial gaze. Entry time is a time from the onset of a stimulus, how much time it took a person to enter a certain area of interest. And over here we've got probably significant difference between entry time to angry face comparing to entry time to happy face. But a red light should light in your head that probably depends on the position of particular face or our stimulus. Therefore, when designing eye tracking study, we present participants with a series of stimulus. And if you got, like in this example, four faces presented on one slide, we manipulate, we change, or we control for the position. So angry face and happy face and sad and neutral, they appear at different corners for different trials. So we can account for the order effect, for example. And also another indicator of eye movement, a sequence. So when we have four areas of interest, which one of them was visited earlier and which one was visited at later stages of the slide exploration. Eye tracking indices from such a study, when you actually test for attentional biases, can be used as indicators of therapeutical change. And over here, you've got an example of a scan path from a clinically depressed individual that was tested before mindfulness training, but it could be any therapeutical intervention. So before, as you see, the participant was mostly concentrating on sad face, a little bit more, less on neutral, and definitely not very attracted to happy face. And this is an example of the same person after mindfulness training, and hopefully we redirected the individual attention toward positive positive emotions, which is, as I mentioned before, not typical for depressed participants to look at happy faces. Analogous procedure, but this time with words as stimulus, we used in predicting daily functioning of depressed participants. In this procedure, we used 12 slides. On each of them, 4 words from different categories were presented. We had dysphoric, aversive, positive, and neutral words. So, and per analogy to the face to 4 faces study. And the words similarly were presented for 10 seconds each of the trial, and we have 12 trials. We look at glances count, so how many times people were coming back to each of the area of interest, and also the main indicator was a dwell time, and we wanted to check what a dwell time on dysphoric words or negative words measured in laboratory has anything in common or has any predictive power to tell us something about daily functioning of depressed participants. Before I tell you about the results, I'll just make a quick note, technical word note. These were the words. And those of you who do not know Polish, you need to believe me that this was a neutral, dysphoric, positive, and negative word. But look, the area of interest is way bigger than the word itself. And this is because when we are measuring eye movements, we are not always very precise or accurate. And it's better to have a bigger area of interest to be able to actually capture the fixations within the area of interest. So the dwell time, or time spent reading simple words, were used as a predictor of daily functioning of the depressed individuals to test the relationship between the time spent on this, looking at dysphoric, positive, or aversive or neutral words. This was measured in the lab. So we had some individual difference because people differ in time they spent looking at these 4 categories of words. And after visiting our lab, participants were asked to maintain a daily diary for 7 days. And for 7 days, toward the end of each day, they were reporting us on their well-being. And among questions that we asked them, there were questions about daily rumination, so negative, repetitive, negative thinking, and also the overall adjustment, how well they think they were doing that particular day. So the data structure was a nested one, and or 2-level data structure. We had a day level, 7 days of measuring rumination, for example, rumination, or in general, day well-being. And at the person level, we knew something about the way they look at they read the words in the laboratory. So in a series of multi-level modelings, we tested the relationship between rumination, what it can be predicted by dwell time on dysphoric, aversive, positive, and neutral words. And here you've got coefficients, which you can interpret as correlation. So, and just for you to remind you that the correlation coefficient can be from -1 to to 1, and the positive correlation, the higher, for example, the more time spent looking on the spheric words, the higher was daily rumination. And even stronger correlation, positive correlation, was found for aversive words. So we may say that the more attention was paid to negative words, the worse was daily functioning of the participants, the higher they were ruminating. And we observed negative correlation between their overall adjustment and again the spheric and aversive words, meaning that the more they looked at the words, the more they focus on the words, the less adjust they felt during their daily functioning. So I think it's very important for pursuing eye tracking studies to find actually the link between something that we are measuring in our laboratories and actual functioning of the press, in this case, the press participants, but you can think of any other groups that you can test. Knowing that we are coming from diverse and interdisciplinary backgrounds, the other example I prepared is coming from rather marketing or advertisement field. I would like to present you a study on consumer confusion, how consumer confusion influence attention to product that are placed in TV shows. And this study is a true experiment, meaning that we manipulated the consumer confusion, and how we manipulated I'll show you in a second. And this could be without the second independent variable, which was within subjects variable. This could have been just a simplest experiment in which you have two groups. And this is an example of over here, our manipulation. So we had a group of participants, which we randomly assigned to two groups. So we divided them randomly into two groups. The first one had a noise video, collection of random scenes presented for 3 minutes prior our culinary program, in which there were different products placed. And the other group had a 3-minute video that we called a calm one. No big changes. There was something that the video was tend to induce calmness and did not definitely produce confusion in our participants. And after 3-minute video with the noise one or calming one, we presented 5 minutes of culinary program, which included different products, names of different, and we were interested in differences in attention paid to these different brands during the TV, the 5-minute TV program, depending on our manipulation, whether someone was looking at the noise of different advertisements, different parts of different programs, or whether person was in a calming condition. I think you can make an analogy to a situation when you're tired and you want to watch something and you just jump from channel to channel and trying to look for something, or there is this advertisement block prior you wanted program, and so the noise condition might be analogy of it. And I also chose this example not only because I think it's a nice experiment, but also that in the study we were showing a short clip, and to analyze the attention paid to different brands, different products, we need to draw or define dynamical ROIs, areas of interest, drawn around the product of interest. So, for example, this product, around it, there is area of interest that will be following this product, no matter how it is presented in the film, at different positions, and even in movement. So later on, we can calculate eye tracking indices, so number of fixations, the total dwell time for this product, and compare it between our two groups. Over here, you've got visualizations along with the areas of interest drawn on the product. Okay, and what were the results? The blue line is for a group that was in the calm condition, and the yellow line is for a group that was in noise condition. And I told you at the very beginning that this could be just a simple experiment with one independent variable, namely the noisy and calming condition. But we decided to slice the 5-minute program into 1-minute epochs and compare how the visual attention to the product placed in the TV show changes over time. And this introduced us the second independent variable that is within subject variable. And what we observed that during the third and fourth minute of the short clip, where the products were in use, we observed significant differences between attention toward these products. The number of fixations was significantly lower in the noise condition. So it's like being fed with something that is cognitively noisy or demanding at the very beginning reduces our resources and leads to cognitive fatigue that was observed in the project or in the result. And I'm just wondering, how are your batteries, cognitive batteries? Are you still following me?
Yes.
Okay. Do you have any questions so far? Except the question for a break. Would you like to have a break? I don't know how organizers preview it.
Yeah, Isa, we can take a break. I don't know, 5 minutes, 10 minutes. How much do you need? It's a good idea, I think.
I'm fine. I can continue. So it's up to our attendees.
We can stretch legs for 5 minutes.
Yeah, 5 minutes
Okay.
would be great.
So let's come back 11:16, and Krzyś, do not use your counter because then we'll meet in 15 minutes.
That's right. Okay, so see you guys in 5 minutes time then.
Yeah.
See you in 5 minutes.
Thank you, Agnieszka, for taking care of questions. I saw that there was a question related to dynamical.
Yeah, maybe you want to elaborate on this actually.
I don't really have to elaborate. You answered correctly, but sometimes if the object is not in move, So you do not have to do it scene by scene, but you do it, but you check when it starts moving or when it's changing its position. And so you draw area of interest there. And then there are some functions that extrapolate the movement. So it's not that painstaking, but I'll show you an example from a study that we've done together with Agnieszka on content and lexical words in subtitles. And we really were grateful to one of Agnieszka students for drawing meticulously areas of interest around each word. But before that, I would like to present result, maybe not result, but rather a design of a study that is completely within subject design, with one tiny exception, but it's just an exception for me to ask you a question. One day, a student approached me asking about designing eye tracking study in which we can test how much attention deaf participants or participants with hearing impairments are actually paying to face. And this was Marta Abramczyk. And thanks to her knowledge in sign language, we were able to actually test participants in quite amusing study, I must say. We tested 23 participants from Polish Association for the Deaf. Most of them, they had profound hearing loss, but we also had, I think, 3 participants without hearing loss. And these were participants who had deaf parents, and the participants were fluent in sign language, and they also stated that sign language was their first language, not the Polish language. And we test our participants, so we treat them as one group. And we ask them to watch several short clips, and the clips were presenting sign language interpreters, as you see over here. And they were telling jokes. And the funny thing about it is that I do not know sign language, so I was not able to tell whether the jokes were good or not, but by looking at faces of our participants, some of the jokes were good because they were laughing, and yeah, I wasn't able to. Although I guess I should learn sign language. I made 2 attempts, but no success so far. And this study was again, in this study again, we draw areas of interest, dynamical areas of interest. This time we selected 2 areas of interest, one around the face, and the face was also moving, and definitely hands were of the sign language interpreter were moving around, and you can see some of the areas of interest were smaller. At some point, they were growing bigger, and that's maybe another hint for you. If you have areas of interest of different size, it's better to calculate proportion or percentage of time spent in an area of interest rather than, for example, just number of fixations to each of the areas of interest. We introduced second or even 2 independent variables that were manipulated within participants. So the whole group saw different types of clips with jokes. And the clips differ in the type of sign language. We had 3 sign language types. The first one was a Polish sign language, which is natural language for deaf participants in Poland. Sign language system. This is like a copy of Polish language, and this sign language is artificial to our participants, and a hybrid that it's quite frequently used would be a mixture of Polish and sign language. But I guess you're in at least some of you know some of you know more about sign languages. So just to briefly introduce the differences between these 3 types. And also we manipulated whether the sign language interpreter was using face mimicry, mainly lips movement, or the lips remained unmoved, and so there was no lip movement. In this case, we created 6 conditions: lip movement present or not, and 3 types of sign language used. And this may be a place to tell you that why it's better to have at least 1 or 2 independent variables within subjects. If we did not have introduced these variables within subjects, it would mean that we'll have to test 6 different groups of participants. So instead of having around 20 participants in total, we would have to have, assuming 20 in group, total 120 participants. Sometimes, and also from the point of view of variability, having 6 different groups of 20 participants, we would introduce variability that comes within our individual differences. When we have one group tested with 6 different conditions, we eliminate the differences due to individual differences. Okay, so I guess this is this is main advantage, one of advantages of within subject designs, that we have we do not have to test so many participants, and one group would be enough, unless we have some between subject variables that we are interested in. So at the very beginning, I mentioned a little bit of the theory of compensation that states that hearing impaired participants, they have probably strengthened visual attention. But if you look in the literature, there is a lack of support for like general enhancement of visual attention among deaf participants. But there are some findings suggesting that they're really good at their peripheral visual field, that especially for the movements, so they will be able to spot movements farther in periphery comparing to, for example, hearing participants. And one question for you comes with the next slide. I'll show you short clips with participants depicted by the circles. And the question would be for you to guess which group of participants is the one that have hearing impairments. And I think this would be enough. Which one is the hearing impaired group, or the group of deaf participants? So I'm initiating the videos. First one. And the second one. maybe I should change the question. Which any one of you, did you understand what the person was which joke the person was selling? Agnieszka? No. Okay.
Not really.
Not really. Okay, so the easy question. First, or okay, on the left or on the right, or the first or the second. Which one is? Do you think the video showing visual attention of deaf participants? Any guesses?
The second.
The second. Can you elaborate? Why do you think the second?
The focus was on the visual expressions, and since the previous slide that said they have better peripheral vision than hearing people, so they can actually focus on the face and actually see the hands at the same time. But the first one, the focus was on the hands. So it was somebody that's not aware of sign language, and they were focusing on the hands to try to interpret what the meaning of each sign is.
Fantastic. Thank you very much for the explanation. And it's true, I a little bit spoiled it with giving you theory before, but easy questions. It's true, partially true. So you are right that this clip depicts how deaf participants were looking at face, and look, the face does not provide any emotional expression, not even lips are moving. So this is the condition of I think system, sign language system without lip movement. Whereas over here, you said that this is someone who is not aware of the sign language, and in fact, these are our participants who had their whose parents were deaf, and they also mentioned in the pre-testing questionnaire that they are fluent in sign language, and they were using sign language as in their work, at least for the last 9 years. So the only difference is that they were hearing participants. So they were this is hearing participants fluent in sign language, but still their visual perception is different, way different than the visual perception of deaf participants. Unlucky for us, we just had 3 participants that were fluent in sign language and were and could hear. And if any one of you would like to finish the project for us, meaning gather more fluent in sign language but hearing participants, we're more than welcome. You are more than welcome, and hopefully we can publish it somewhere. So yeah, changing the topic, with eye tracking you can also test children, and in this project we had 2 natural groups being compared. We compared children that were starting their music education at the very beginning of the primary school, when in Poland you can so everyone starts primary school around 6 or 7 years old, and some of children can enter music school, so it's like professional music education. And we were trying to figure it out whether the fact that you are trained in music, in reading notes and rhythm, and also theory about music, changes anything in your cognitive functioning. I saw a question during Pivy's keynote about cognitive abilities and eye tracking, so maybe partially you will find answer here. We used mixed design. As I mentioned, we had music and non-music children as a between subject independent variable, not manipulated. Luckily for us, because that was the question or remark or concern expressed by reviewers of our paper that it should be manipulated, but it's not that easy to convince some of the parents, to take part in an experiment and experiment with your child, to send your child to music education. Although being a parent of 3, I can tell, and 3 who went through music education, it wouldn't be a bad idea. The second independent variable was within subject, and this is the type of a task we asked participants to do. And also we test our participants, children, we follow them till the 3rd year of their education. So we started at the very beginning, they entered primary school, then after a year of education, and at the beginning of at the end of the 2nd year of education. So actually we followed them for 2 years, not 3 years. This is the experimental procedure that we ask our children to follow 3 times. And we measure their ability to inhibit oculomotor reflex with anti-saccade task. The task probably is known to our psychologists who participate in the summer school, but might not be that known to other participants. In this task, we ask children to indicate the side of on which side of the screen a target appears. The target is simple dot. Every trial starts with a fixation point. So at in the middle, we present a dot, then a cue, an arrow pointing to a direction at which target will be presented, or pointing at the direction at which target will not be presented. In the task, you've got prosaccadic trials, and these are congruent trials, meaning that the arrow is pointing to in the direction at which the target will appear. So these should be easy trials, but there are also anti-saccadic trials. And in these trials, there is no congruency between cue and the target. So if the cue is pointing to the right, participant already should know that the target would appear on the left. And we are measuring how easy it is for participants to suppress the automatic prosaccadic movement toward the direction pointed by the arrow and switch to the other direction to actually notice the target. Here is an exemplary scan path. Easy question for you, again, from a prosaccadic or antisaccadic trial. Just to remind you, circles represent fixations, arrows or lines represent saccades. Prosaccadic or antisaccadic trial? Are you still with me? Don't make Agnieszka answer all my questions.
Yeah, this one's too difficult. Yes, please, someone help me.
Again,
we have 50% of chances, so prosaccadic.
So I'll explain the task again. Thank you, Marta. So we start with a fixation point. Then there is an arrow pointing to a direction, so pointing to the right. And in prosaccadic trials, the cue is pointing to the direction at which target appears. So in prosaccadic trial, an arrow pointing to the right and target appears to the right. Whereas in the antisaccadic task, the cue is misleading. So pointing to the right for example, but the target appears on the left. Okay.
I would say it's like anti-saccadic, no? Because otherwise you wouldn't actually go back and look in the other direction, right?
So thank you. So it was Agnieszka then. 1 point for you, Agnieszka. So everyone starts from the very beginning. And okay, so this is one example from one person. So this person starts from the middle. Then there was a cue, probably pointing to the right, and the person, a child, made this prosaccadic movement, but then came back. Okay, so this is an example of antisaccadic trial. And since also think about your potential eye tracking projects, sometimes it's enough if you can tell whether a person was looking to the right or to the left. So your areas of interest can be quite big, and this was the case in our situation. So we just like divided this entire presentation screen into two, and we measured the time, for example, of a first fixation or the latency of first saccade toward the target. You do not have to be, if we imagine that we make really tiny area of interest around this dot, we might not capture all fixations that children were making. And because to be able to do it, would have to have really precise eye trackers and also very good calibration. I'll tell you a little bit of about calibration and calibration error toward the end of the talk. So what we observed in time, in terms of response time, we have measurement at the very beginning of the school, after the first first year and the second year. And so what is the general pattern for music school and for general school in a response time? So correct response time was getting smaller. So children with coming age, time passing, were faster in actually pointing to where the target was presented. So this would be a general pattern, but if you look at the trends, we observe that the linear trend for music school was significantly steeper. So the changes in reaction time were faster in music school. And you might ask a question whether it was due to initial individual differences. We controlled in our analysis fluid intelligence. So we took into account the potential differences in intelligence between children, and we still observe significant differences in reaction time. And we were wanted to make a link between the way children were looking during the task, so between eye movement analysis and the reaction time. And what we observed is that the changes in response time were mainly due to the fact that the latency of first saccade toward the target was getting shorter and significantly shorter in the music school, and we did not see this improvement in the general children. So if we come back here to the exemplary scan path, we measured latency of the first saccade toward the target. Latency meaning how much time it took them to correct for the wrong prosaccadic movement and come back with their eyes to the target. And this is an indicator of ability to control something that is reflexive, and because the prosaccadic movement is a reflexive one. So we believe that we observed a significant increase in oculomotor control among children from music schools. We also asked these children, but we did not differentiate this time between music and non-music school. We asked them to read short sentences, and the sentences were differing in one word, in a keyword. And this is typical what you observe in reading studies, that you present a sentence. This is, sorry for Polish, but we studied Polish children in Poland. But still you can recognize that there are some words. And the words were from 4 categories. We could have longer words, so long words versus short words, like over here, this is a long word comparing to a short keyword. And also the type of a keyword could differ in terms of whether the word is frequent or infrequent. So we've got these 4 categories. We had long and frequent words, long and infrequent words, short and frequent words, and short and infrequent words. And we compared aside of traditional eye tracking indices, meaning the number of fixations or the duration. Even by looking at the visualization, you can tell that when a word was long, the keyword was long and infrequent, comparing to long and frequent word. So unknown long word took a little bit of our children cognitive resources to be processed, and they made longer fixations, and comparing to short and frequent words. So 2 fixations would be enough probably to get the meaning of the whole 5-word sentence. We wanted to check whether there is a link between word difficulty and cognitive load. And to do it, we look at the eye tracking indicator that it's newly in every study, I guess, gathered, pupil dilation. And this is thanks to Professor Duchowski's idea. He introduced new measure of cognitive load, LIPA. And LIPA is computed as a proportion or ratio of low to high frequency of pupil diameter, changes in pupil diameter. And it's expected to decrease with a higher cognitive load. And this is what you have here. So depending whether the word was short or long, of high or low frequency, we can observe differences in this measure of cognitive load. And when the frequency, there was this main effect, that the frequency of word made a difference. So when a word was of low frequency, then this indicator was lower, suggesting higher cognitive load. I hope smoothly to move to reading studies, and if you are planning to run or you're doing readings of various kinds of reading studies, what are the units of analysis? In our project, we had a series of sentences, 5-word sentences, as I mentioned, differing in keywords, short, long, frequent, infrequent words. And each of a word can be a separate area of interest. And for each of the area of interest, then we calculate eye tracking indices, meaning number of fixations, the length of fixations. Sometimes there are more than one fixation for a word, so we can calculate total viewing or total reading time. And depending on what is your main goal of the study, you can either analyze the whole sentence. You can analyze separate words, as in our case, there were keywords, but there are also studies on, for example, optimal placement of fixation. So you can study just the beginnings of the words, or even syllabus or letters, whether the recognition of a word or the coding of word is easier, whether depending whether your fixation is placed on a consonant or on a vowel. And also you may present the whole paragraph, like in a study in which we compared dyslexic and non-dyslexic children. We can, so there was a paragraph presented, but you can do the analysis on the whole sentence or the whole paragraph, or that it's quite common way, as I mentioned before, you can treat each of the words separately and then categorize the words into according, for example, to the number of syllables that the word have, or the frequency or the function, whether this is a function or lexical word. Over here, you've got samples, 2 sample scan paths. Just you can watch it, and we try to observe all different phenomenon that you can analyze during eye tracking reading studies. So number of fixations on each word. Like, for example, over here you see 2 little fixations, 1 longer. Regressions. So we tend to come back to parts of a text that we do not understand or we got lost, or you'll see we might have different difficulties in controlling eye movements, and might have different difficulty in precise placement of fixations. So we can observe regressions, refixations, and if a word is easy or we don't want to really read it, we can just skip it. And there is a whole bunch of studies showing if neighboring effects. So that the fixation, duration of fixation, the dwell time on a word depends on neighbors. So if the preceding word is a short one and easy to process, then there might be some preview benefits. So with your periphery, you can decipher or decode part of the word of the next one. So the neighboring effects, preview benefits can be observed. Even if you know if you know a word from the context, you can skip it. So let's try to find all the phenomena that you can observe during reading. I stopped for a second. Look over here, the first sentence. Most of the time we just, in reading, if we are fluent in reading, we just move from one word to another, we move forward. So 50% of time during our reading, we just smoothly go forward. But sometimes we make refixations and come back. There is no regression yet, I guess, here. Oh, there was one regression. Difficult word to process over here. Even difficult to pronounce. You can try pogwizda. Even for Polish people, pogwizda is difficult to be pronounced. Someone asked about individual differences. This was a study when we compared dyslexic and non-dyslexic children during reading. And I don't even ask a question because it's too obvious a question. The bottom scan path was presented by a child with dyslexia. And what you could observe here, lots of regressions. Typically 10% of our during our reading, we make regressions. Around 20% of the time we refixate, and the refixations are related to processing difficulty. And also around 20% of time we're just skipping words because either they are supportive words, we can say, like function words, so they function around lexical words. They do not have a meaning by themselves. Different types of reading. You can also look at how children are reading music notes, and then treat group of notes as words, and these would be more difficult. In this task, children were asked to tap the rhythm of the notes. Okay. I mentioned that now I can see Agnieszka smiling. She recognized this scene from our study on reading subtitles. And I already mentioned, I think, painful work at some time, or very boring work that was done by Agnieszka's MA student, who draw areas of interest around each of the word in a subtitle, so we can later find out differences between function words, attention to function words, and attention to longer words, lexical words, differences between people with hearing impairments and without hearing impairments. And if I recall properly, but Agnieszka can correct me, we observed that deaf participants made devoted surprisingly quite a lot of time to reading function words, which are typically skipped by fluent readers. And to us, it meant that maybe Polish, and it's true that Polish language is not under native first language, so they their eyes behave differently than the fluent reader, who would typically skip the function words. But I'm not mentioning this study because I would like you to get interest in the results, but to show you how you can analyze film with subtitles. So one way would be to analyze the subtitle and the content of subtitle, but you can just draw one area of interest around subtitle and compare it to attention devoted to the rest of the scene. You can also analyze, and this is what we've done also in one of our study, analyze transitions between different parts of the visual field. In this case, another clip with subtitles, and we were interested how much time, how many times, or how disturbing is a shot change to reading subtitle, and whether after a short change people are coming back to the beginning of subtitle, checking whether something new, a new subtitle appear because the change of a scene. So we had three areas of interest: image, beginning of subtitle, and the remaining part of the subtitle. And you can examine transitions with transition matrix, transitions between different parts of the scene. So knowing, I try to explain how to read this transition matrix. So we're starting from, for example, image. So the first fixation on an image, and what is the probability that if I'm here somewhere around, I will stay there? Around 60%. If I'm at the image, do I move to beginning? Okay, so from image to image, from beginning to beginning, and from rest to the rest of the subtitle. So you can have compound images, and then as I gave you the example with for faces or for words, and then you can analyze transitions between different between different areas of interest. Coming to the end of my talk, or the last part of the talk, is not the end yet. Don't get too excited. Oops, it's going to be recorded. A bit of technical information. The projects I'll told you about we run them in our eye tracking research center, which is not that old, does not have that long traditions as PIV Center, but still we have quite a nice selection or range of eye trackers, and a little bit of the last part of my talk will be devoted a little bit of to a selection of eye tracker for your future projects. I'm not telling about the project during summer school because, as you know, it will be an online project, and you will find out more about how to do it and what is the platform by our next speaker, Adam Salary, who will introduce you to this platform. So altogether we have 24 video eye trackers, which I think is quite a good density, knowing the size of our lab. 11 gaze points, or total 15 gaze points, because we have more like for didactic reasons with lower sampling rate. And this is something new that I'm mentioning, sampling rate, and I hope some of you knows what I mean. The rest will know after the talk. How many times per second eye position is tracked. So if you have 60 hertz, 60 times per second, we know where the position, we check the position of eye. And the higher sampling rate, the more accurate, the shorter eye movements we can measure. So we have 4 gaze points with 150 hertz, one SMI, which I a little bit nostalgic about it because this is my favorite eye tracker, but apparently they got sold and no support is provided. But the eye tracker is still working. So, but I made this decision to put it to the box. Eye tracking is moving out of lab toward the wild or into the wild. So we do not restrict our participants to sit in front of a computer with the use, for example, of Pupil Labs eye trackers. And I'll present a short clip with potential with one of the applications. So we have 7 Pupil Labs allowing us to test collaboration between our participants. And at our university, we also have an eye tracker, which I like to call a Mercedes, but Krzyś is laughing at me when I'm doing it. Is an Mercedes.
Or a Tesla now, perhaps.
Tesla. Yeah, let's be pro-ecological Tesla. So the Tesla eye tracker iLink has possibility of tracking the position checking position of eye 2,000 times a second. So you can imagine how precise little eye movements we can analyze with it. In terms of different eye tracker providers, you saw Pivy wearing ad hoc eyeglasses, and they claim they can be used the whole day. So you can imagine more studies with tracking participants' activities during their daily lives. We also have a project on maybe going into classrooms where both teachers and students can wear such glasses, and we can analyze how much time they're actually paying attention, teachers to their students and students to the content of the talk. Are you still with me? Without eyeglasses, I'm just checking.
Just regular glasses on.
Regular glasses, yeah. So I mentioned Gaze Point. You'll hear a talk presenting this type of eye trackers, but we recently exploring biometrics. So measuring heart rate variability that is aligned, that is synchronized with eye tracking data. One of the, and this is a little bit ironic because Tobii is one of the most popular eye tracker providers. They have all different kinds of eye trackers. And this is the one that we are missing in our lab. And eye tracking is also, as you saw with the keynote by Pivy, going virtual. So we can also track participants' eyes while they're playing some kind of very immersive games. I know also that I think it was Tobii that they designed an eye tracker that goes along with a gaming set to help gamers to interact with their eyes, I don't know, shooting with their eyes or selecting objects. So they using gaze as an input to build more immersive, entertaining environments. And so this is also a possibility to run studies in these virtual environments. And I mentioned eye tracking in the wild. I would like to present you with a project, I think very interesting, showing how we can collaborate and how we can track participants' eyes during collaboration. And this is a project by PhD project by one of our tutors or hands-on helper, Kasia Wisiecka. In this project, you will see a brief video on it, but we used Pupil Lab eye trackers and two people collaborating on large screen. And over here you can see, or maybe it will be better on the second one. You can see our markers that are used to indicate what are the areas of interest that for which we could then analyze eye tracking indices. So I'll show you a brief clip, and I hope you can, adjust the sound that you will hear it properly. So just Agnieszka or Krzyś, because I can see you. Let me know whether you can hear it properly.
No, there is no sound. Could you make sure you are sharing the sound? It's one of the...
I did not make sure that I share my sound. Hi, can I make sure that I'm sharing the sound?
You look into the Zoom options, you should be able to see either on top of your screen, like rolling out the menu, or at the bottom with the green button. Can't remember right now because I'm not sharing at this moment.
Okay, I can see audio settings.
Can you go to share screen?
Enable, okay, I click something which is enable original sound. Maybe.
No, I think it's about screen sharing rather than audio. Because like on the in the
Wait, I can only stop screen sharing, that's for sure. I can do new screen.
and at the bottom you have this green share screen icon.
Yeah.
And if you click on the little arrow on the right, do you have the option to share sound?
No, I can just make a new share, but you know, what we can do, we can have it share sound. Okay, maybe we'll check it right now.
Decision-making with large-screen collaboration, evidence from an eye tracking study.
Collaboration on renewable resource management can enhance sustainable decision-making, while system dynamic simulations, or SD simulations, sometimes involve two players. The interfaces have so far not been specifically designed for multiplayer collaboration. Comparing in-person, remote, and single-user setups, we examined how these setups can help improve problem-solving strategies. Our research employed advanced methods, e.g. eye tracking, to quantify collaboration on SD simulations through measures of task load, collaboration, and usability. Besides this, we also attained design-relevant knowledge for SD interfaces. Our paper presents empirical findings based on time series data, participants' answers to questionnaires, and eye tracking data. Our research addressed 3 alternative setups for simulation of natural resource management: single user, in-person collaboration, and remote collaboration. We hypothesized that collaboration leads to more effective task solutions in simulation. We also theorized that visual attention distribution differs between experimental conditions, which may influence decision-making strategy. Our experimental procedure and setting was similar in all 3 setups, with a few minor differences given by the nature of the remote setup. The single and in-person setups use the same basic version of the simulation interfaces. Here, we see the single user setup in action. And here, with the same interface, we see the in-person setup in action. The remote setup required remote players to log in to the gaming session. They then went through the same instructions as usual, plus remote-specific instructions on an accept-reject mechanism, which was activated in this setup. Here, we see the remote setup in action. The simulation user interface was equipped with areas of interest visual markers, AOIs, for the purpose of our research. AOIs were not visible to the participants. AOIs marked with blue color relate to simulation control. AOIs marked with orange relate to decision making. AOI markers help capturing and analyzing eye gaze data. Based on eye gaze data captured, here we created heat maps presenting attention distribution for each of the 3 experimental conditions: single user, in-person collaboration, and remote collaboration. Here, we see accumulated simulation performance data. Herd size outcome is shown left, lichen density outcome is shown right. Color coding is the same for herd size and lichen density. The optimal solution, plotted in gray, is the outcome from the model simulation. It shows the maximum sustainable reindeer herd size and lichen density. Experimental run 2 is plotted in orange. It shows mean participant performance. For more results, read our paper.
Okay, do you hear me?
Yes.
Okay. So this was an example of 3 types of collaboration, and I hope that you see the potential of the eye tracking moving away from the goal lab. There are various applications for eye tracking in the wild. We know collaborators from Belgium who are interested in sports psychology, and they, for example, explore the differences between experts and novices in various disciplines of sport, tennis, badminton, but you can even imagine having study while biking to your work, and you bike with eyeglasses to tell us scientists to which parts of the environment around you pay attention.
Supporting
Okay. The last part from my talk from me, a little bit about precision and accuracy. So getting more technical, let's say. So this is a typical image you would see from eye tracking camera. I don't know whether you recognize any similarities to one of the summer school organizer. And so we video-based eye trackers, they use illumination with infrared illumination to make nicely visible pupil, and the sparkle in the eye is the reflection from retina. And I'll tell you how it works. You can get different, even more creepy examples of the camera image. Over here, this is the pupil and reflection from the surface of your eye, and the image reflection sometimes is called Purkinje image, and we tracking with video-based eye tracking the relationship between the reflection and the center of the pupil. The corneal, that's the reflections that it's named. It's called corneal reflection, and it's relative to the pupil, to the center of the pupil. And when we move our eyes, the corneal reflection changes position, and this is how we getting. We can then estimate where we were looking. And as I mentioned, the corneal reflections sometimes are called Purkinje images after Czech physiologist who is recognized as a founder of psychophysiology, and he was the one who first noticed that the light reflects from our eye. So how the eye tracker works. So we capture eye image by the video camera. We've got the eye in the light, in the infrared light for better pupil distinguishability. So we are able to actually detect the pupil, and then the relation between pupil and the reflection tells us something about the position of eye, the direction in which we're looking, and we're mapping the coordinates of the visual scene onto the camera image, and this is how we're getting the eye estimation. With the rapidly changing technology and new ways of capturing eye movements, this might change, or this is different, but this is definitely true for video-based eye trackers. And we would like to be precise in the way we capturing participants' eye movement. And so we would like to reduce calibration error. And every eye tracking study starts with with a calibration procedure in which we testing the precision of eye tracker, and also we would like to get the coordinates and make sure that the participant is looking where we would like him or her to look at. So, for example, this is a target. And this is where the actual gaze point where we are looking. And we would like to minimize the error. And the error is typically expressed in angular degrees, and we would like to be precise around half of a degree. we might expect different scenarios with our accuracy and precision. We would like all the eye trackers to be in this bull's eye, in the center of the bull's eye. So accurate and precise, but we might also end up with something which is not accurate and not precise, so we cannot trust our data. What I mean by saying accurate is that it's close to the target. So our gaze is close to the stimulus that we ask participants to look at. We may be very precise. So whenever we're looking at something, we're looking at certain space, but the space is far away from the actual target. So when you'll be selecting eye trackers for your use, it's good to check what the providers say about the accuracy and precision. And as I mentioned before, we start our eye tracking studies with calibration. So we ask kindly our participants to move around the screen to follow a dot, and typical standard calibration is a 9-point calibration. And you've got an example over here of a good calibration and not very, and the one that we would wish probably recalibrate or would not, and this is quite often that we, if we do not have good calibration for a participant, we repeat it once or twice, but sometimes it's hard to get good calibration, and these are our individual differences, I guess. Over here you've got an example of calibration errors. So the gray points are the point where a point appears on the screen, whereas the cross is a place of our gaze. And as you see, everything is in green, meaning that the calibration error is small enough to be accepted. And as I mentioned before, half of an angular degree is a good calibration, but we also accept calibration with one degree of difference. And I guess this is probably all what I'm going, what I were selected for you to hear from me. Your projects during the hands-on session will be done on online platform, Real Eye, and I'm not telling anything about it because you will have the whole talk by Adam, I guess just after lunch. And I just wish, or I can't wait to see visualizations of your online eye tracking projects. And I hope I'll be helpful, or we all be helpful to in designing your first eye tracking online projects. Maybe the projects will not be first in terms of eye tracking, because I know that some of you already have some experience, but I guess that there might be first online eye tracking projects. So thank you very much, and also thank to our team. Without them, the projects will not be possible to run. Thank you, and I'm happy to answer any questions you've got with the help of Agnieszka and Krzyś, of course.
That's right. Thank you so much, Isa.